What I'd like to start with is by reminding what most of us know, which is that the embryos are the great masters. Throughout evolution, they have discovered those exquisite ways to form that are not only fascinating, but are also very useful to solve real-world problems. We can learn enormously from them. Almost everything is there. In our case, we are studying the early mammalian embryo that is called the blastocyst. It is a powerful model, not only because it comprises all the cells that will generate the full organism, but also because it is autonomous, it is free-floating, it is adaptive, also we can also say it's self-organizing, and finally it's small enough to be studied in great detail. And the biggest problem that we have studying embryos is that they are extremely difficult to work with. They are tough nuts to crack, and if we want to really and deeply understand them, I would argue that we must be able to build them from their individual components. And the structure that you can see here is in fact remarkably similar to the mammalian conceptus just before implantation into the uterus. At that stage, it is called the blastocyst. It comprises about 100 cells, and part of it is going to form the placenta. It's the outer red cells that you can see here, while the rest inside will form the fetus and other extra-mammalian tissues. However, this specific structure that you see here did not arise from the fusion of egg and sperm, but it actually formed through self-organization of stem cells all in a dish. So this is our approach to rebuild the embryo to better understand it. And before we start, I wanted to recap some key vocabulary and knowledge that will be necessary for the rest of the talk. So what you see here is an oocyte that is being fertilized by a sperm to form a genetically unique zygote. And this zygote in the mouse and in the human is going to divide and form 2 cells, 4 cells, 8 cells. And during those first divisions that happen during the first days, you can see the days of the mouse development here. During those first divisions, the cells are thought to be very similar to each other in terms of potential. The system is also very plastic. You can remove one cell and still form a full organism. However, when the embryo, the conceptus, is reaching the 16-cell stage, there's a very clear event that happens where the cells in the outer are becoming one lineage that is called the trophoblast lineage, and that will form the full placenta, while the cells inside remain totipotent for some time, and then very quickly become pluripotent in order to form what will become the fetus. And once this first lineage segregation has occurred, the structure can form this blastocyst, which is our stage of interest, which is formed with a fluid-filled cavity that you can see here, that is surrounded by an epithelium of trophoblast, and that shelters this inner cell mass. And this inner cell mass is going to form 2 cell types. First, those red cells that are called the epiblasts that are going to form the full fetus. And then a second cell type that's called primitive endoderm that's going to form the yolk sac. And at this point, it's ready to implant inside the uterus. But what we can do is to harvest those blastocysts and put them in culture. And depending on the condition that you use, you can expand the stem cells that it contains. So of course, if you, as we all know, you can expand the embryonic stem cells using LIF, leukemia inhibitory factor, but you can also expand trophoblast stem cells using fibroblast growth factor 4. And these are stem cells that can be massively expanded, they can be genetically edited, they can be extremely useful tools to study development. And they are frozen in time, and the best way to show that is that you can re-inject them into an embryo, and they are going to participate to the development of the embryo itself or the placenta. So what we did in the lab is to combine those two stem cell types and to promote their self-organization into a structure that's remarkably similar to the blastocyst, and that I called a blastoid. And of course, this allows to do a couple of experiments that we could not do before, just for the sake of producing them in large numbers, but also because we can do a couple of other things with them. So this is brief summary. Again, we can get the blastocyst, form, extract and derive embryonic stem cells, trophoblast stem cells, expand them, genetically edit them, do all kind of interesting things at this step, and then after reaggregate them and form those blastoids. And in order to form those blastoids, we have to first deposit the embryonic stem cells. They are going to form a cluster here, and then we sprinkle the trophoblast stem cells on top of them, and they are going to form a double layer structure that, under specific conditions that I'm happy to discuss with, are capable of forming a blastoid. And those blastoids are forming very efficiently if you have the right number and ratios of cells. If you have about 8 embryonic stem cells and about 20 trophoblast stem cells, the efficiency is about 70%. This was quite an exciting time when we published this in 2018, because for the first time we had a model of the full conceptus. We had a model that represented both the embryonic and all the extra-embryonic compartments. And beyond the morphology, those blastoids form analogs of the 3 lineages. Here you can see the trophoblast analogs that are in red and that are normally going to form the placenta. You can see the analogs of the epiblast in white that will normally form the full body, the epiblast, and the third cell type, the primitive endoderm, that will ultimately form the yolk sac. And those primitive endoderm cells are forming by differentiation from the epiblast. And because blastoids are modeling the pre-implantation stage, they could be transferred inside the uterus of a pseudo-pregnant mouse where they implanted. And you can see here, so you can see here on the left horn, we transferred the blastoids. On the right horn, we did not. And what you can see here is that the uterus is reacting by forming a cocoon around it. This cocoon is called a decidua. And within a few days in utero, those blastoids are growing. The structure is elongating, the cells are differentiating, but very quickly, the cells are getting disorganized. And we have not formed a mouse from blastoids. Actually, the experiment has been repeated now, and nobody has formed a mouse out of a blastoid. But the reason why we formed those blastoids is that by observing such embryo models forming, we can interact with previously inaccessible principles of development. And especially this is powerful to highlight what are the minimal requirements for development to occur. The famous clause of necessity in science. What is necessary for the embryo to develop? And obviously we don't have at the moment everything to have those structures developing, but we are using this bottom-up approach in order to understand those minimal factors. And if we can better understand and answer questions with blastoids, it is because early mammalian embryos come in very small numbers and remain difficult to experiment with. Despite the CRISPR breakthroughs and all kind of other ways to manipulate them, they are still difficult to manipulate. On the contrary, the blastoids can be formed in virtually infinite numbers, and they are also amenable to high-throughput genetics, incorporation of complex circuits, or they are amenable also to molecular screens. And these are some of the important bases for scientific and biomedical discoveries. So using this model, we have focused on re-exploring a very classical concept in developmental biology, which is called embryonic induction, when one group of cells is directing the development of another one. And the classical model of embryonic induction was depicted for blastocyst development by Cope. It was in 1978, and he suggested through correlative observations that the blastocyst form through molecular inductions that are originating from those inner epiblast cells and that are driving the development of the outer trophoblast. Cope was actually using a microdissection to isolate, to cut the blastocyst and to isolate the left and right parts, and by growing them separately, he proved that the inductions are locally regulating the trophoblast proliferation. And it's only 20 years later, in 1998, that the lab of Janet Ronson in Toronto showed that a key molecule that acts as a mitogenic inducer is actually FGF4, fibroblast growth factor 4. And that is actually the rationale behind using FGF4 in order to derive mouse trophoblast stem cells. And for a long time, this was actually the only known inducer. So we caught up this on this concept and tried to fill the knowledge gaps here. And we actually did this by systematically comparing blastoids with trophospheres. And trophospheres are formed exactly in the same way as blastoids, but without the embryonic stem cells. So we deposit the trophoblast stem cells in those microwells that you might have seen in the previous slides, and we use the exact same medium in order to that we use to form blastoids, but we do not put the ES cells. And you can see already that the trophospheres are quite different as compared to the trophoblast of blastoids. And this already tells you a little bit about what is the role of those cells. And of course, the first thing that we want to do is to check if the model is recapitulating the classical experiments of Cope and Tanaka. And so we look at proliferation, and what you can see here is that we start with a certain number of trophoblast stem cells, and during the course of blastoid formation, this number is clearly increasing. So those cells are proliferating. On the contrary, trophospheres barely do. So in exactly the same conditions, you can see that the presence of the embryonic stem cells is clearly extremely important to regulate what was known as the only embryonic inductions. And so this is interesting for us. We see that the model is actually recapitulating what is known, and that clearly those inductions are extremely important for building the blastocyst and the blastoid in that case. And a second thing that we can look at is a second function that is classical for stem cells, which is self-renewal. So we can do a colony formation assay, isolate the trophoblasts from blastoids and from trophospheres. And you can see here that the trophoblasts from blastoids have a much higher potential to form a colony as compared to the trophoblasts of trophospheres. So this tells us that those embryonic inductions are not only regulating proliferation, but also the self-renewal of those stem cells here. Whereas in the absence of those, they are losing this capacity, they are actually differentiating. So this is good. We are recapitulating what was already known, and clearly the hypothesis was very strong. And now what we can do is to systematically observe the role of those embryonic inductions by comparing blastoids and trophospheres. And this is what we did here. This was early days of single cell sequencing. What you can see here is a UMAP of single cells that were taken from either trophoblast stem cells that are cultured in 2D. So this is the starting point. You can see them here. Also embryonic stem cells that are cultured in 2D. And then you can see the trophoblasts that were coming from blastoids. And these are actually embryonic stem cells that are coming from embryonic bodies that are cultured exactly in the same condition as blastoids, but without the trophoblast. And now you can see here the transcriptome of single cells isolated from trophospheres. And this is actually embryonic stem cells isolated from blastoids here. And now we can systematically look at the embryonic inductions by comparing the transcriptome of this group of cells and this group of cells. And we came up with a couple of bioinformatic analyses. I'll just show you one, which is related to signaling pathway activity. And you can see that at the top of the list, we get actually like the MAP kinase pathway, which was very important for us to see, and this is confirming the work of Tanaka in the lab of Janet Ronson, that FGF4 is indeed produced by the epiblast and is regulating the MAP kinase pathway into the trophoblast. Now we can go down the list. There are all kinds of extremely interesting things. Just can point at the Hippo pathway, some classical developmental pathway, JAK-STAT, the Wnt. And we actually stopped at the end of this list on the TGF-beta pathway, because there were some interesting candidates, nodal and BMP4, that are being expressed by the epiblast. However, the functions are not known, so we can now do a couple of assays, and I'll just show you one here. Where we, in fact, look at a very simple morphometric features of blastoids and trophospheres, which is the diameter of the overall structure. And so this is the diameter of a blastoid. We are actually looking at more than blastoids here. We are looking at everything that we had in the plate, but the average is here. And you can see that the average of a diameter of a trophosphere is actually smaller. And now what you can do is to replace the embryonic stem cells by the factors that they produce, and to dose BMP4 or nodal on top of those trophospheres. And what you can see here is that the diameter is increasing quite nicely for BMP4, about 20% increase for nodal. And when you combine the two, they are actually like they add up. And the second thing that we did after looking at this is to count the number of cells in all those trophospheres. Actually, the number of cells are absolutely flat. So those molecules are not regulating proliferation of the trophoblast, but they are allowing for the structure to still increase in size. So what we hypothesize at this point is that those molecules might play a role in order to maintain the epithelial integrity of those trophoblast cysts in order for them to swell. So for a structure to swell, you have to seal it, and then you have to pump. And what we suspect is that the BMP4 and nodal are acting on the sealing. And so we worked on this hypothesis by first doing knockouts for nodal only in embryonic stem cells, an experiment that is quite difficult to do still in the blastocyst. And what we observed is that indeed, like the size of the cavity is decreasing. So we are phenocopying the phenotype that we saw by dosing a nodal on top of trophospheres. We can then sequence those structures, and what we find out is that there are two key transcription factors, CDX2 and KLF6, that are regulated. We're quite happy to see this because CDX2 knockout has been described, and they actually form a blastocyst, but the blastocyst cannot expand. The epithelium is too rough. It doesn't really like form properly, and those blastocysts cannot implant inside the uterus. So we thought, okay, this is fitting quite nicely, but this has been described already. So we are going to look at this KLF6. KLF6 has a defect, but the defect that has been reported is much later during placentation. And we also saw a bunch of cell-cell junctions, tight junctions, and adherence junctions that were genes that were regulated. So what we can do now is to do a KLF6 knockout only in the trophoblast stem cells, and then we can form the blastoids. And what we see is that we phenocopy the phenotype where the size of the cavity is decreased. These are two different clones. So this is coming to this idea that we have like nodal that is being secreted by the epiblast, and that this is regulating the epithelial development of the trophoblast, and that if you don't have, and that this development is partly regulated by KLF6. And this is quite interesting in terms of embryology, because it shows that the effect of a mutation into one of the lineages can actually propagate to the other compartment. And so this a mutation into one compartment can actually deeply change the whole organism. And these are hypotheses that we can test now. And it would be actually interesting to know if the size of the cavity that we decrease here is actually impacting back into the epiblast by reciprocal inductions or other mechanism. It is possible that the size of the cavity changes, for example, the local concentration of proteins or metabolites is affecting the embryo itself. And this allows us to think of how embryos might have evolved and change their overall size in order to develop different functions. So the last experiment that I'd like to show is that we can actually go back to trophospheres now, and we can stimulate those trophospheres with BMP2 and nodal, and the diameter is increasing. And now when we do the same thing with KLF6 knockout trophospheres, you can see that the trophospheres are smaller, and then they don't respond at all with BMP4 and nodal. And we found a couple of things that are not properly regulated and that are related to epithelial development. One of the things is keratin 8, that is quite well known to play a role in epithelial development, and ecaderins, also the intracellular part of ecaderins that was downregulated. So this is interesting because by comparing this transcriptomic data, by generating knockouts in functional assay, we can fill this knowledge gap and show that the inner epiblast is not only producing FGF4, it's also producing other factors. I didn't show you, but it's producing IL-6, it's producing IL-11, producing nodal, BMP4, also BMP7, which are regulating different trophoblast function. And the first big function is growth. By promoting both the proliferation and the self-renewal of the trophoblast, those inductions are maintaining the stem cells that are going to sustain the growth of the placenta. And this proliferation and self-renewal is mostly regulated by FGF4, IL-6, and IL-11, according to our experiments. By regulating those pathways, you are regulating self-renewal proliferation. You can allow for the placenta to grow on that side. And so this is the first function. And the second function that those inductions are regulating is a function of patterning. The combination of proliferation and epithelial morphogenesis, that is regulated by nodal and BMP4 at least, allows for the blastocyst to form a cavity. And the main function of this cavity is to distance a pool of trophoblasts from the inducers. And this is this pool of trophoblasts that is being less exposed to those embryonic inducers. And as a result of this distancing, these cells opposite of the cavity are rapidly differentiating, and they become sticky. And this is this stickiness that allows for the blastocyst to attach to the uterus. So you see that the second function of patterning, that is combination of proliferation and epithelial morphogenesis, is extremely important in order to form this other pool of cells that is going to play a very crucial role during the interaction with the uterus. And this is a wonderful picture of a mouse blastocyst that has just landed and attached to the uterus lining, which is called the endometrium. And this picture was taken by the lab of Anne Sutherland, and it captures the first touch between the mother and the embryo. And as you can see, it happens through those sticky cells that are opposite of the epiblast of the. So one last experiment that we can do with the mouse is to see if those embryonic inductions are actually necessary for the implantation inside the uterus. So we can transfer them back into the uterus. And what you can see here is that the trophospheres have a strongly diminished potential to interact with the uterus. And so we verified here that those trophospheres that lack the inducers they are barely proliferating. They are barely self-renewing, they fail to undergo proper epithelial development, and they differentiate prematurely. And when transferred into the uterus, they have lost the capacity to interact with the uterine tissues. So this is kind of interesting if you look at the big picture here, because as we all know, like those extramedullary tissues, the placenta and the yolk sac, are extremely important to support the development of the fetus. But at the very beginning, before implantation, it is actually the opposite. It's the embryonic part that is going to guide the development of the extraembryonic tissues. It's going to shape the development of those trophoblast tissue, and the reason why it's doing this is to allow for this tissue to implant inside the uterus. So in some ways, at the very beginning of life, it's actually the future fetus, the epiblast, that is investing into its own future by fueling the development of those extraembryonic tissues. And of course, those tissues are paying back 1,000 times later by nurturing them for their later development. A couple of years ago, we thought that it would be nice to be able to also tackle those questions in the human system. And the reason why we wanted to do this is to test whether those same basic principles are occurring in the human. And because there's one interesting difference between mouse and human, which is that their blastocyst implants via their opposite side. So in humans, these are the sticky cells. In the mouse, I told you that the sticky cells are here. In humans, they are exactly like 180 degrees on the opposite side. So sometimes during evolution, the system underwent a 180 degrees rotation, and this rotation probably had very important consequences because it strongly constrains the way that the embryo is further developing inside the uterus. A second interesting difference that we thought would be good to look at is that the cavity of the human blastocyst is actually proportionally larger in human than in mice. So when thinking of the roles of those loops of inductions between the embryonic and extraembryonic lineages, those facts are resonate here. However, at this point, no one knows, and partly because of the difficulties in studying the human blastocyst. So in order to form human blastoids, one has to start with the right cells. And we found out that by aggregating those so-called PXGL human embryonic stem cells that were established by the lab of Austin Smith at that time in Cambridge University, and by stimulating them with the correct cocktail of molecules, and here we are talking about a triple inhibition of the Hippo, ERK, and TGF-beta pathway, human blastoids can actually form with a remarkably high efficiency. And this efficiency is not anecdotic. When you see such a efficiency, it gives you confidence that you actually you're starting with the right cells and you're stimulating them in the right way. We actually achieve about 70% of efficiency, which was quite spectacular for us. And so using all those different cell lines, we always have about 70% of the microwires that are filled with a structure that is morphologically similar to the human blastocyst. And that means the cyst, a cavity, and one unique cluster that forms on one side. During the formation of these human blastoids, there's a very interesting process of growth and specification control that happens, by which the system is sensing and adapting its own size and the ratios between the three cell types that it forms. And we are actually currently investigating these so-called self-organizing phenomena. You can see that the cell numbers are quite tight and between the three cell types, and that the ratios are actually even tighter. But before we engage into any type of mechanistic studies, it is extremely important that we properly evaluate how closely human blastoids are mimicking development. And on these UMAPs, you can see that over time, the PXGL human pluripotent stem cells that are in orange here are first generating 2 cell types within 24 hours. And this one cell type is very close to the PXGL cells, and it's the epiblast of the blastoid. And the other cell type is further away, and these are the trophoblast. And only in the second step, between 60 and 96 hours, the epiblast is producing a third cell type that is an analog of the primitive endoderm. And this is the precise sequence of blastocyst development. Trophoblast first and primitive endoderm last. And this whole process happens within 96 hours, which is quite similar to the timing of human blastocyst development. Between the morula and the late blastocyst, there is about 3.5 days. And this is extremely important because we have like a sequence and a timing that is similar to blastocyst development. And importantly, at the end of the process, we have only 3 cell types that formed. These are actually controlled that we spiked into the experiment. These are so-called primed human embryonic stem cells that represent perigastrulation stage embryo. And these are the human trophoblast stem cells that were derived by a Japanese group study led by Okai a couple of years ago, and that represent a post-implantation stage. And you can find back, these 3 cell types that are clearly segregated. Their transcriptome is clearly different, and each of them expresses the genes that are specific to the 3 lineages. Here we have genes that are specific to the epiblast, SUSD2, a very classical marker, OCT4. And here you have some that are classical to the trophoblast, GATA2, keratin 18, and here classical for the primitive endoderm. However, this is not sufficient for really like understanding if at the transcriptome level those cells are similar to the blastocyst. If we want to do so, we have to merge those blastoid cells with cells from human embryos that are harvested at different stages. And here you can see in blue pre-blastocyst stage human embryo. Here the blastocyst, which is in green, you've got the trophoblast here, the primitive endoderm, and the epiblast. And here in those 2 colors, you have the post-implantation stages. And you can see that the cells, so this is the reference map, and the cells of the blastoids are very nicely matching with the 3 different compartments of the blastocyst stage. We have spiked primed human embryonic stem cells that are very nicely hooking up to the gastrula stage. And overall, we counted that we have about 3% of the cells of the blastoids that are off target. And a lot of them are clustering with the extra-embryonic mesoderm of the gastrula stage. So overall, human blastoids are forming efficiently, timely, according to the right sequence, and they are forming about 97% of blastocyst-like cells. In 2021, there are about 6 labs that proposed protocols to form models of the early human embryo. And the single cell sequencing RNA sequencing data from 4 of them have been reanalyzed by an independent consortium that was led by Sophie Petropoulos in Montreal and Frédéric Lanner in the Karolinska Institute. And from this analysis, it appeared that forming blastoids with blastocyst-like cells requires that one starts with the right shade of human pluripotent stem cells, and we are proposing the PXGL state as a very good state. And we have to use the right cocktail of molecules. And if not, the system can form morphologically to some extent, although at low efficiency, but it will comprise mostly post-implantation like cells. Of course, because cells evolve rapidly during development and to fulfill their different functions, it is very important to form the right cells for the model to be predictive of human development. And here you can see the timeline that Frédéric Laller and Sophie Petropoulos put together. So you can see the pre-implantation blastocyst stage here. The blastocyst starts on day 5. It goes over day 6 and day 7. Here you've got the trophoblast that form, here the epiblast, and there's another, the third cell type, the primitive endoderm that forms here. This is another reference data set, but what we can see here is that the blastoids are very nicely clustering with the primitive endoderm, the epiblast stage, the primitive endoderm stage, and with the trophoblast stage. These are the naive cells to start with, these are the primed cells, and these are the human trophoblast stem cells that I told you about that are also post-implantation. So this part is post-implantation trophoblast. So this is the molecular characterization is very interesting and important, but we also want to know if those structures have a specific functions. And of course, because it is ethically unacceptable and now actually forbidden by the International Society of Stem Cell Research to transfer human blastoids inside the uterus, we developed an implantation model assay using endometrium organoids. So we can expand those organoids. This is done in matrigel, and it has been done by 2 different groups, the group of Margarita Turco in Cambridge and the group of Hugo van Kelkom in Belgium, in Brussels, in Leuven, excuse me. And so we can expand those organoids, and then we can plate them in 2D in order to facilitate the deposition of the blastoids. And then we can stimulate those with the hormones of pregnancy, progesterone and estrogen. And those hormones are triggering the receptivity of those endometrial cells. And very interestingly, when the endometrial cells are not stimulated with hormones, the blastoids can literally sit on top of them for days without attaching. There is no interaction that occurs between those cell types. On the contrary, upon hormonal stimulation, before we deposit the blastoids, this is priming the endometrial cells, which allows them to attach the blastoids. And you can see the blastoids, we have about like 30, now we have more than 40% of the blastoids that are attaching, and they start to grow on the polar side, on the side that is close to the epiblast. And this is exactly what you actually see during early human embryogenesis. So there is a very specific interaction that is happening between the blastoids and the hormonally stimulated endometrial cells. And beyond this, in this movie, you can see that before attaching, the blastoids orientate and position themselves with the polar side down. And this is not anecdotal, but this actually happens very robustly. And this is suggesting that the blastoids are forming those sticky cells that we were talking about previously. And that they are forming them at the right spot, which allows for the adhesion to the endometrium. And you can see here on this movie that that was taken just after just at the moment when the blastoids are attaching. We are pushing the medium around the blastoid with a pipette, the pipette that you can see here, and you can see that they really attach very nicely via those sticky cells that are abutting the epiblast. This is suggesting that somehow the blastoids are forming spontaneously on axis. So we looked at this axis formation, and we observed that the majority of the blastoids, about 60% of them, are undergoing a process of trophoblast maturation in the polar region, that is marked by the expression of NR2F2, a transcription factor, and by many other genes that we pull down by RNA sequencing. So this is suggesting that, contrary to mice, in humans, the embryonic inductions that are coming from the epiblast, that you can see in yellow here, are not only promoting the growth of the stem cells that are forming the placenta, but they're also inducing the maturation so to make them sticky and allow them to attach to the uterus. We had about like 30% that did not form an axis or did not form an axis yet, and we had a very small percentage that form an inverted axis. So if the hypothesis is right, by preventing the formation of the epiblast, we should be able to impact the maturation of those polar trophoblasts and their stickiness. And we actually prevented epiblast formation in two different ways, to form what we call trophospheres, just like in the mouse system. We did this by inhibiting the STAT pathway, or by it's actually over-inhibiting, very strongly inhibiting the Hippo pathway using XMU-MP1, a small molecule. And those trophospheres, we analyze them by transcriptomic, and they are actually reflecting the early, those ones are reflecting the early and the late stage trophoblast. Blastocyst trophoblast, and accordingly to the hypothesis, those trophospheres are actually incapable of interacting with the hormonally stimulated endometrial cells. So this is fitting with this idea that those epiblasts are really like inducing the local maturation. And we actually added two controls here. The first control are human trophoblast stem cells that are post-implantation, and those aggregates of human trophoblast stem cells are also incapable of interacting with the hormonally stimulated endometrial cells. This is a bit of a silly control here, but it's just more technical control. We also made aggregates of naive or human embryonic stem cells. They are also incapable of interacting. So this is actually like quite striking, because if the cells, if the trophoblasts are too early, they are not capable of sticking to the endometrium, and if it's too late, they also have lost this capacity. So clearly, those embryonic inducers are creating a short window of opportunity for the embryo to interact with the uterus, and this is actually a concept that is quite well known in clinical embryology. It's called the window of implantation. So we think that we might have recapitulated this clinical concept of window of implantation. And of course, now we have the possibility of like finding the pathways and the processes that are behind this. So through a transcriptomic analysis, we actually extracted a handful of ligand and receptor pairs that might be mediating this initial interaction between the embryo and the mother. We can also culture those human blastoids for longer time on top of the endometrial cells, or also on plastic, and we did this until a time equivalent of 13 days. But of course, these are not reflecting a human embryo on day 13. However, the blastoids are capable of very nicely expanding the three lineages. So the three lineages are progressing and they are proliferating. However, this has very little to do with a day 13 human embryo. So I'd just like to conclude by we are now in a position to try to understand how during evolution those two species have created partly different ways to form, especially how this axis has inverted and led to the embryo implanting via diametrically opposite side. And it is tempting to think that this opposite mode of ingression into the uterus had some important repercussions on the early development of those two species. Of note, like the first weeks of human embryogenesis are very prone to failure. there are about 50% of human fertilized eggs that fail to form a baby. And among those 50%, 75% of these pregnancies are failing around implantation time. So this implantation time is a bottleneck of human development. And how and why such fragility of early development appear during evolution is actually unknown. And although it might have contributed at some point to the fitness of humans, it is currently causing all kinds of problems of infertility, pregnancy failure. It also has a deleterious impact on the adult health. Those first weeks are actually extremely important for later, much later events. So I believe that the fundamental understanding of the specificities of early human development is actually providing a powerful lever to improve global health through effective family planning, reducing an ongoing global fertility decrease, and also maybe preventing diseases in adult life. Finally, I'd just like to finish by saying that we have to communicate properly about those embryo models. At the moment, those blastoids are very rudimentary, and they are clearly very different from embryos. For example, the epigenetic imprinting, the X chromosome status is extremely likely to be very different. And we have to recognize this. So similar to a Picasso painting, we can kind of recognize what is depicted, but we also have to admit that this is quite different from the real thing. And if you are interested in the ethics of embryo models, you can have a look at these papers that we have partly participated to, and would like to thank the ISSCR for taking us seriously and for developing a nice ethical framework for this research. So with this, I'd like to thank the people who participated to the human study that we did at the IMBA Institute of the Austrian Academy of Sciences, and especially Haru Nobu, that you can see here, Alok Javali, and Haydar Haydari, who, along with Teresa Sommer, who was a master student, who was a PhD student in the lab, have developed all this. And I'd like also to thank some collaborators, especially Laurent David in France, who has done the reference data, a lot of work to get reference data set for the transcriptomic of human blastocyst, and Hugo van Kelkom in Leuven, who helped us with the endometrium organoid. I thank you very much for your attention.
I may take the chairman's prerogative and just ask you to perhaps speculate a bit about how you might use your system, and this is a pretty obvious question, to actually start modeling diseases. You have the great advantage of being able to look at the earliest stages of pre- and post-implantation human embryogenesis. We believe that many diseases, even those that sometimes don't become apparent until later childhood or even adulthood, begin at that point. Can you envision how, or maybe you've already started doing this, how you could start modeling certain diseases? Like you alluded to pre-implantation, problems of pre-implantation, but maybe even pregnancy-induced hypertension and the preterm birth and things of that sort. Have you started pursuing that?
This is extremely interesting. So these are very early days, and it is not completely clear when are those defects or insults happening that would impact development and adult life. However, from epidemiological studies, it is clear that it is the first weeks of pregnancy are extremely important. One of the interesting facts is that there's an epidemiological study in Netherlands where they looked at the weight of babies based on at the end of the Second World War. And they actually, like in the last year of the war, there was starvation in Netherlands, and the starvation led to babies that are smaller. And those babies were especially smaller when the starvation happened in the very, very first months, probably the first 2 months of pregnancy. So, and very strikingly, 50 years later, there was a surge of chronic diseases that happened in Netherlands. So from those same children. So there's clearly a link here, but and the link is thought to be centered on placental development. So the development of the placenta in the early stages seem to be extremely important in order to develop a proper connection with the mother, a proper turnover of the vascular system of the mother, and in order to fuel the development of the fetus. And if this does not happen properly, the first thing is preeclampsia. And of course, this leads to very often to pregnancy loss. But if this happens suboptimally, then the placenta cannot develop properly, and this impacts directly the development of the fetus. So, for a very long answer to a very good question, I think the remodeling of the vascular system by the trophoblast in the first weeks is probably like a very important event that we might be able to model in the next few years.
One of the questions is an interesting one about experimental method. The questioner was wondering, obviously blastoids are asymmetric, but the questioner was wondering, could you artificially generate a symmetrical blastoid by using opposing differentiating gradients, since it's an artificial system?
Yeah, so this is we are definitely trying to do this, because, especially in the mouse, because you can, by manipulating the symmetry, you can manipulate the way it interacts with the uterus. And so this is something that can be done. It can be done using externally imposed gradients. It can also be done by developing some synthetic biology tools so that the regulation of the left and right part of the blastoids happen in different ways. So they are complicated experiments, but I think we are extremely insightful about what makes the cells sticky on one side and what actually maintains the stem cells on the other. And these are absolutely key. We are trying to publish some of this work now. I hope you will enjoy it when it comes out.
Great. The next question is, why do humans have low implantation success? Is there a natural quality control process to guard against using implantation as a quality control system?
No, but this is an excellent question, and this is a question that we can tackle now. So we are very interested in this. So why do humans have low implantation success? It can be because of the embryo itself, and there are a lot of clues that a lot of human embryos are aneuploid or comprise aneuploid cells, and those aneuploids we don't know exactly why and how they occur, but they occur quite early during embryonic development. Probably during the first divisions, and they are detrimental to the further process of development. So that's for the embryo side, and for the uterus side, there could be defects in the attachment to the endometrium, and that would be a defect into, for example, the formation of sorry, I'm going back to the embryo side, but it could be a defect into the formation of the sticky cells in the embryo, and it could be also a defect in the lining of the uterus, the endometrium. In mice, there's also strong clues that the decellularization process that happens after the initial attachment is prone to failure in aging mice. So aging mice are losing the capacity to form the cocoon around the blastocyst, which leads to termination of the pregnancy. So all kind of questions. We are in the position of tackling some now. We are building up on to answer the next ones. But yeah, one thing we cannot answer all the questions now. I think the question of attachment are clearly possible. The questions of destabilization, we are going to have to build a more complex uterus organoid.
We noticed that one of the molecules, one of the signaling molecules that you pinpointed early on was IL-6. IL-6 is also well known as an inflammatory cytokine that results after very viral infections and other types of pathologic processes. Do you envision any way to make a connection between some of the congenital problems that can happen during development and viral infection of the fetus in utero?
Yeah, so what is clearly known, so there are two aspects. There is IL-6 in the context of development, and here it is activating the STAT pathway. So this is regulating the STAT pathway into the trophoblast and also into the epiblast, and this is leading to classical developmental cascades. And besides this, of course, it has a role into regulating the immune cells. And here it turns out that the immune cells are extremely important for the implantation process. At the moment of implantation, there are immune cells that are recruited, natural killers and others, that are here to clean the decidua from senescent cells. And this cleaning will allow for the embryo to invade. This is one function quite well known, but there must be like many other ones. And it is becoming very clear in the last decade that the immune cells that are recruited are extremely important for this implantation process. So this is clearly something that we could add into the uterus organoid at some point. So we need to go step by step, but there's a strong possibility that we can recapitulate some more processes here.
Yeah, and I guess we're getting down to our last question, and it was, in your system, do you yet see the emergence of the products of various germ layers, for example, mesoderm, ectoderm, which would give rise to vasculature and nervous system, or is it too early in your system to start seeing that?
So it's a very good question. So we haven't looked at it, but we are culturing. So a blastoid is reflecting a day 7 blastocyst. Then we culture those for 6 more days. So we have a time equivalent of day 13. As I mentioned, this is a time equivalent. This is not a structure equivalent. We think that we are probably around everything is a bit asynchronous, I believe, but we are definitely not at day 13. As you know, like the gastrulation happens in the human embryo on day 14. So we are just before this. And of course, there's currently an ethical debate to whether we should culture human blastocyst for more than 14 days. And we argue that human blastoids are only embryo models and are not the equivalent of human blastocysts. So there's not necessarily a restriction for the culture of human blastoids, but because the debate is ongoing and because we don't want to create any unnecessary controversies, we actually stopped the culture on the time equivalent of day 30.
Great. Well, thanks so much, Nicolas. That's I think we've run out of time for any more questions, but this has been stimulating and fantastic, and we look forward to seeing exactly where you'll take this great model system.